phagocytosis. In the previous lecture, we had seen the various procedure of phagocytosis, that is how phagolysosome is formed, how lysosome can lysosomic enzymes are responsible for the phagocytosis. Now, what are the complements? Complements are proteins which are present in the body. It is a cascade of reaction. What do you mean by cascade? Cascade means what? The activation of first compound or for activation of first protein is responsible to activate the second protein, or the activation of the first product is responsible for the activation of the second compound to give the product. So, this is chain of reaction. Okay. So, the most complement protein is C3, which is present over there. What we are going to say, this is the microbial activation of the alternative pathway. How microbial, this is the cell surface of the microbial, and this complement system is result in the flow of sodium, potassium and from outside to the inside, so that the internal environment of the microbial is get changed, or there is the lysis of the membrane. So, after this, what we are going to see is what? This is the last product of this pathway, that is solutes are come inside. This is the cell surface. Okay, so, this is a microbial cell surface, this is the inner side of microbials, and from where the solute concentration is start increasing. Okay, so, this is the what is this? This is the we can say that the result of complement pathway. So, two main proteins C3 and C5. The C3 is present in the blood or present in the plasma in the inactivate form. Okay. So, microbial polysaccharides, do you remember that in the previous lecture we had seen microbial polysaccharides are the polysaccharides which have the role in the system or role in the immune system. Now, this C3 is get converted into the C3b, okay, and C3a. It means what? The C3 which is present in the plasma, it undergo the proteolytic cleavage, or in the presence of water, it reaction with water, it produce two fragments C3a and C3b. Okay. C3 is the chemoattractant to eosinophils, okay, responsible for the eosinophil chemoattraction. Now, C3b, once it forms in the presence of iron, magnesium and another factor C3b, it get converted into C3bBb. Okay. So, first what is first? First C3 get converted to C3a and C3b. Second, what is this? C3b is a product of C3, activated form of C3 get join or form the complex with Cb in the presence of magnesium ion and form a big complex C3bb. Okay. These undergo the cleavage by factor D and produce a complex who has C3 convertase activity in such a way that this C3 convertase is responsible to maintain the concentration of C3a and C3b. Okay. So, C3 is responsible, so that is C3 produce C3b by this enzyme and reaction with water, but there is again the recoverance of C3a. Okay. So, C3b and Bb produce the C3a. So, okay, what is this? Is a C3 convertase activity of this complex in the presence of porphyrin. So, C3 and C5a both are important in this complement pathway, which are small peptide generated from the parent compound, that is C3 and C5. Okay. So, that is during the complement activation. So, in this first part, that is microbial activation of the alternative pathway, where we learn that there is a generation of C3a and C3b. Okay. The cycle is such that the concentration of C3 and C3b will maintain, because here it is required. What is this? This is the requirement for the formation of the complex. What it is termed as the membrane attack complex, MAC. Okay. Once the C3A and C3B is formed, it attack on the C5 and C5 convertase convert that C5 into the C5A. Okay. It means that generation of this C3 and C3B and its association on the microbial surface convert that into the C5 convertase, so that when this complex is linked with C5, it will convert it into the C5A. Okay. So, what is this? This is a inactive form. This is the enzyme convertase which convert the C5 inactive into the active C5A. And what is this C5 convertase? It is a C3B and BB. From where C3B and BB comes? It will comes from this one. Okay. So, in this way the C5 is get converted into C5A and C5B. Okay. So, what is the role of this is C5A? See, this C5A is the neutrophil, or you can say that it activate on the phagocyte, it activate on the neutrophil and it stimulate the respiratory burst. What is respiratory burst? It means that it stimulate the formation of reactive oxygen species and reactive nitrogen species, so that it will start degrading the biomolecules which are present over there on the surface, means cell surface carbohydrates, cell surface glycoproteins. So, this reactive oxygen burst will responsible for that. We had seen that mechanism in the last lecture. Okay. So, now C5A get form attached to the over here, and C6 and C7 both make a conformation change in C8 and it is responsible to get insert into the microbial surface. C9 undergo the conformation change to the active form, and C5B and C9 both together are responsible for the formation of membrane attack complex at the cell surface, so that there is a leakage from the membrane will start. Okay. Now, this leakage, what it is termed as? It is termed as the post C3 pathway for the production of what is this? MAC, membrane attack complex. Okay. Now, how this C6 and C7 is responsible? What is the function of C6 and C7? The C6 and C7 will get joined to the C5B. Okay. So, once they will get joined to the C5B, this complex has two functions. C5B plus C6 and C7 come over there, they form the complex. Once that complex is formed, this complex has the affinity to C8, and so that C5B, C6 and C7 together attract the C8 over there, and this C8 has the ability to get bind as a transmembrane binding site. What do you mean by transmembrane? Means what? It has a site or it has a tail and head which is on the cell surface, and the tail is in the cytoplasm or in the below inside of the microbial okay. Now, what are C8? What is the form of C8? C8 is in T form, alpha chain, beta chain and the gamma chain. The alpha chain of C8 has a capacity that it will direct lead to the conformation change in the C9 protein and convert it into in such a way that in the active form. In such a way, that means what? C9 will get into its active form. So, once it will get into the active form, this has the capacity. It is an ion amphipathic molecule, and it get inserted into the lipid bilayer of the membrane. Okay. It undergo polymerization, it form a ring like structure, and from which the solute are come from outside to the inside and from inside to the outside and disturb the internal environment of the microorganism, and there is the lysis of this cell surface will start. So, in this way